Okay, let me know screen is visible.
Yes, ma'am, it's available.
Okay, so from today we are going to start our new module, that is R for bioinformatics. Are you aware about R programming used before?
No, ma'am. Not yet.
Okay, so fine, no problem. So first, in today's session, we'll understand what exactly R, how it is useful to handle biological data, how to install this, what are steps, how to work with this. Okay. So from next session onwards, we'll see actual scripting. So first thing, yeah, we are going to cover these all points. R programming for genomics, R genomics and proteomics both, like how R in data science, then introduction of R in bioinformatics, what is the means why it is a basic language and what all basic syntax are required to execute this language, application of R, and hands-on session we'll see. Okay, so first I'll like to tell you one thing. When we execute any code, means any functionality of R, it is divided into three different classes. Like first thing, when we want to summarize, as if we consider as a role, different roles. So as a bioinformatician, there's a different role: statistics application or statistical analysis, and data science. But all three roles, like what role exactly they are doing. First thing, we need data. So data interaction is first important thing. After getting data, we need to analyze that data. So analysis steps are there. So this is a statistical analysis. And then after analysis, the most important thing, that is a interpretation. So interpretation we can do by using visualization, by plotting different graphs, charts, and different visual plots. So this is three steps: data interaction, analysis, and result visualization. So if we consider as a data science, so data science in the context of bioinformatics, so this is just like simply we can say it is like a art for extracting knowledge, and we can fetch information, that is a biological information. Okay, so nowadays this is like scope, like interdisciplinary, we can say interdisciplinary approach. So we can examine this. R is very powerful language, and we can find mysteries hidden, information like in genomics, proteomic information. So R is very versatile and open source language. So let's see why it is important in data analysis, like specifically biological data. What do you think, why it is required, R for analyzing biological data, or any programming language? As any
Because we have
biolog,
a huge amount of data.
sorry.
We have a huge amount of data.
Yes, we have huge amount of data. That is the main thing. To handle this huge data, we need software, need analysis, we need algorithms. So that's why we need this all programming, or this all well structured language. Okay, like genomic data, it's very complex data. Proteomics data having complex information. So through this effective data analysis, we can decode different patterns, we can identify different trends, and definitely we can draw meaningful conclusion. Okay, so advancing our understanding of genomics, proteomics, and definitely we can contribute some groundbreaking discoveries by using these all things. Okay, so let's see some theory part, like what exactly the R in bioinformatics. So as I told, R is a powerful programming language which is widely used in bioinformatics for analysis, statistical analysis, or data visualization. And definitely it provide versatile environment so we can handle complex biological data. Then analysis capabilities means what? So like most of the data we handling in Excel file format. So our Excel these files, and we can use different algorithms tools for analysis. So different robust statistical function we can apply to process, to interpret the data set, large data set we can say. So we can find essential information from this genomics and proteomics study. For this, what we require some packages. As in Python we used like pandas, numpy, matplotlib, biopython. So similarly in R there are many packages which are corely focused for handling biological data. Okay, so we can say like these are specialized package which is made. So for example, if I can say this is a bioconductor package which is very useful to handle biological data, like suppose you want genome data analysis, sequence alignment, or any bioinformatics related task we can perform. Then visualization. So visualization libraries are there like ggplots, which enable the creation of detailed and informative graphs, and definitely help to present bioinformatic data clearly and effectively. So visualization is important part of the study to interpret the result. Then integration with bioinformatic tools. So integration means we can work with different tools, databases on the same platform. So yeah, definitely workflows or any efficient data processing, data analysis. To work with this, it is easy with the R platform, R environment. Now let's learn some basics of our programming language. The most important thing, we should understand our syntax which is understandable by that environment. So we have to write code in such a way that programming language can interpret. So syntax and structure, that is in R it is like we can use different assignment operators, functions, control structure. To understand the structure is a main part, like essential for effective coding we can say, and analysis interpretation. For that, we need uh, data types. To store that data type, we need data structure. So data structure here R supports various data structures like vector, matrix, data frame, list is also there. So each structure serves specific purpose. Like it allows researchers to organize the data, manipulate the data efficiently. So that's why structure is important. Then function is again integral part of R. So we can facilitate the task, suppose a simple calculation or any complex algorithm if you want to perform. So by using function we can minimize the code, because repetitive task we can do just by creating function or by applying function. So again, like Python, there are two types of function: built-in functions and also user defined function. User defined means we can create a customized function, so depending on the experimental requirement, so it definitely enhance our versatility. Then again, libraries are like packages. So tidyverse is there for visualization, ggplot, hr for normalization, downstream analysis of NGS data. There is a dsec2. So these are different specialized function and tools which are used for manipulation, visualization, and statistical modeling. So definitely it helps us to solve this code. And okay, we'll see application in of BAN-R in bioinformatics and genomics. So specific application in genomics, like when we need to handle complex biological data, a biological database like any data, genomics, proteomics, or we can say omics data. So researchers can efficiently prepare, pre-process, and analyze, and after analysis, interpret the large and intricate data set. Okay, then visualization, again the same point is there, like pre-processing from raw sequencing data to identifying, suppose we are focusing with a variation data, so genetic variation or any analysis of gene expression pattern that we can visualize. These are specific applications. Statistical analysis. So statistical analysis, like suppose genomics data we have and we want to do task like differential expression analysis or variant calling, or just want to find the correlation, identifying the correlation with different data set, comparison. So these are all things we can perform. Then machine learning nowadays it is very required. So when we want to implement such kind of algorithm, learning algorithms, like there are many models are there: predictive modeling, classification, then clustering is like a pattern recognition. So in genomics, in proteomics, we can do this all algorithm, we can take help of these algorithms and perform. So definitely it contribute in things like personalized medicine or biomarker discovery. So these are all advanced part in R. Along with some benefits, there are some challenges as well. So the main challenge is like, as sir told, dealing with large scale genomic data set. So genomic data analysis, these are some unique challenges due to scale and complexity of the database, and definitely it is generated by, nowadays we are using this advanced sequencing technologies, so we are getting raw data, and this is a big challenge. So dealing with large scale genomic data set, like large volume of genomic data, so data that might be whole genome sequencing, transcriptomics, then whatever the application we are running, but whatever data we are getting, how to store that data, because we need some structured organizer after storing, whenever we want to process that data or analyze that data. So if it is stored properly, then by applying different algorithms we can process or analyze that data. So this is a one complex uh, challenge. Then complexity and heterogeneity. So as this data is very vary, like DNA data, RNA data, someone working with epigenetic data, so there is a heterogeneity in data, various data types, and because of this we require integrated analysis approach. Computational and storage demand. So computational and storage demand for all this processing of genomic data, like resources we need, then efficient algorithm infrastructure we require, and that is a big challenge. So importance of efficient tools. So tools like packages, different packages for manipulation, analysis, visualization. So R as already a powerful statistical programming language, but still they are providing different packages, but there are some challenges to advance these all packages for advanced use. Then parallel computing and optimization. Parallel computing definitely enhance the scalability of analysis, and we need to do this and application, reproducibility and documentation. So reproducibility for what? Like if we maintain reproducibility in analysis, it's critical because most of the time our code matters transparency, documentation support, or sometime reproducible research practice will be there. So again, this is a challenge faced in this omics data analysis, particularly handling large data set. So definitely we need to explore what exactly the significance tools, overcoming challenges, then some rules to follow these all things. Okay, now we'll see how to install this on your system. So for that, you all can open browser. I'll give link in chat box. Okay, so this link, when you will click on this link, it will open page, this page, posit.org. So just click on this, I'll guide you so you can install. So R is a free software and software environment for analysis, statistical analysis, statistical computing, graphics. Okay, so when you click on this, you'll get these two options: install R and install R Studio.
Ma'am, can you please again repeat the step where we have to click? Hello.
Yes,
Ma'am, can you please repeat the step where you have clicked just now?
I'm not clicked anywhere. You just open this page. I'll first explain. First we need to install R. Okay, and then we will work on R Studio. So I'll explain what exactly the difference between these two. First we need to install R, so you just click on this link, download and install R. So when you install, when you click on this link, it'll give you this page. Okay, so according to your operating system, like the distributions are given: download R for Linux, Mac, and Windows. So as my system is Windows, so I just click on this. When I click on this, there is an option base, as we need base like software install R for the first time. Okay. So when you click on this, it'll give this link download R. This is a latest version 4.4.1 for Windows. If you click on this, you'll get .exe file and your downloads. So just wait for installation and let me know after completing. So this what we are installing, this is a R base. R base, or we can say R software. Okay, so our software basically when we install on your system, on your computer, this is your personal computer resource. Means you can execute or process this R programming language. And R Studio integrates this R as a IDE. So IDE is what? Integrated Development Environment. So we can take different functionalities from this R Studio. I'll give this. I'll explain this when we install that R Studio.
So it's already downloaded. Shall I continue with the installation?
Yes, yes, yes. You'll continue. You'll get a message like, "Are you want to run this software?" So you just need to just click all this process. Just keep all default setting.
Okay.
Request permission will be there to install this on your system. So select that and preferred language. I'll prefer English. So there is a drop down list for language. Then just you just need to follow the steps like next and agreement. You have to just click on next. These all steps will be there. You just proceed with this and then finish last. No need to
Ma'am,
customize any option.
I have to save it in C program only, no? C file.
Yes, yes, by default all software save in C program, so don't change any path, don't do any customization option. Just do whatever the default one. And there will be a option to generate a program shortcut, so click yes. So that shortcut will generate on your desktop, because as like as all software when you install, the shortcut will generate on a desktop, so we can access that software. So shortcut. After all steps, you'll get one message like installation has completed, and just to click on this finish button for
Yes,
complete the
I'm
steps.
done.
You
Ma'am,
take your
here we
time.
have
Yeah.
to click on the create the desktop shortcut.
Yes, yes.
Okay.
So this base R is basic software which contains your R programming language. And R Studio is also software, but that makes R programming easier by IDE. Okay. That is more user friendly, like graphical user interface. You can just click and proceed with any command. No need to type every command.
Um, should I download our studio?
Uh, yeah, you can download. No problem. Done with R base.
Yes.
Both of you? Okay.
Yes,
So how
ma'am.
to search like our base is installed or not? Because if we search here, as it is a software, so definitely shortcut will create. And let's check. Okay. So here is the software. Okay. Just click on this. So such kind of window will open. You can open all of you. So I'll explain how to operate with this. So as this is a software, this is called as a core software environment of our programming language, and it provides all fundamental functionalities, or you can say provide all core features of our program. But this is a command line interface. Command line means what? You have to type command, and through command you can give command directly work as a R console. So like for interaction with this R base, suppose I want to perform anything, like here I'm writing 2 plus 3, this I want to perform. So what can I do? I'll just write as a command and then execute. So writing command on the console itself and executing in the same area. So this is R console. Okay. So means this is for beginners, it is easy, like you can execute write command and execute here itself. So this is like a direct interaction with the R. The script there is no separate script editor. This console itself acts as a script editor. So here you have to write, edit, run everything. Okay. Now here you can open new script if you want. Then from file you can access these all options, like if you want to save history, load history, then these all options. Edit for editing, paste and all things. Then visualize, toolbar, status bar, packages, like from this package you can set. I'll explain these all things so you'll get a clear idea when you compare with our base and our studio. So this is a cran. Then repository you can select from here. Install package. So if I install packages, if I'll click here, so you need to write the from cran which package you want. Here itself you can mention install dot package, like this command install. And there is no what we can say, auto detection. Okay, so you have to type code by yourself, like this. If I'll write in bracket, I need to write name of the package, whatever package we want to install. Okay. So here there is like syntax highlighting or code completion, error checking, and some enhancing options are not here in RBase. So this is all about RBase. If we want to use plot, like if you want to generate any plot and then visualize that plot, so graphical output, basically you need to create plot and charts, and then whatever generated graphics by using plot function, like here we use install packages. Similarly, you have to write this plot straight, and whatever dataset you are working with, that dataset you have to provide as an argument inside this plot function, and you can visualize the directly through this command line. Okay, this is all about our base. Now we'll see what exactly our studio. So you can install now the second option. Here it is. The same way you have to proceed. This download our studio desktop for Windows. You can proceed with this option. And after installation there will no shortcut will generate, and this will create as an app. So if you want to search after installation, you have to open like this R Studio. Such kind of app will be exist after installation, R Studio app. So you need to click on this app. Okay? Just let me know after completing this.
It's done on this side.
Okay, fine. Uh, yours, Sanjana?
No, ma'am. 2 more minutes, ma'am. It's now also downloading.
Okay,
Okay, so
let me share screen. I'll explain. Let it download. Okay. So once you download, just search in search box at the bottom left bottom side for R Studio. So you can see the app and just click on that app. So this is your R Studio, that is integrated development environment. Okay, now let me explain this. As there are total 4 panes. Okay, we in R base what we saw, console. Okay, but in R Studio, this is a IDE and specially designed for R. So if you can check this, it is very user friendly interface with the different tools and different features for R programming. So when you see, I think your console is at the bottom left bottom side. By default it is at the bottom left, but if you want to set or arrange as per your convenience, you can just switch between this console and environment. Like here you need to check session, sorry, view. So in panes, basically each region is called as a pane. So in pane, you have to switch console on left or console on right. By default, it is on left, bottom left side. If you want to check all this layout, there is an option at the bottom, pane layout. So you can check what all tabs you want to see. By default, whatever it is you can see in this IDE. Okay. So this is for customization option. So now let's discuss this area, 4 different panes. So the first important, that is script editor. This one, script editor, basically when you want to write any script or edit, execute. So if the same script, if I write here 2 plus 3, now when I execute this, either you can use this tab run button or you can use shortcuts. It is written here control plus enter. When you click here, you'll get the shortcut. Run the current line or selected, so you can also select multiple lines. Otherwise, where cursor is blinking, you can use that line for execution, or by using control enter you can execute this like so. This is just for writing script and your execution will be in console. Okay, now if I'll write the same command install, so here what happens, here there is a code completion. When you type only install, it will show the recommendation install packages and all. So definitely it helps when you means there is no mistake of spelling or writing and all, or it will give you a recommendation for all commands, whatever the pre-built, like from PRIN there are many options, so you can select which you want, which one you want to execute, like this. Now this is for script. So here you can open multiple scripts just by clicking this new file plus sign, or you can also use file option, new file, R script, or this is a shortcut, control shift and any one option you can use. So this way, if I click here, it will open new untitled one new script. Here also you can write multiple script, you can open and execute simultaneously. And suppose here I have created one object. As we already learned object of variable concept in Python, I am writing some value, assign some value for this object. So when I execute this object, so in environment, that object is created along with the assigned value. Okay, in console it is executed. Okay, like by print statement, if I want to execute this value, so I'll call this function and call this variable as argument, so I can execute this. So this is script area, this is console. Now
ma'am,
let's see.
excuse me.
Yes.
Ma'am, for execution, are you clicking on something? Means after completing the script, are you clicking
I'm
on something?
using shortcut control plus enter, or if you want you can use this run button.
Oh,
Like click here, this cursor is blinking, right? Let me maximize this. This cursor is blinking, you just need to click run. Otherwise if you click here, you'll get this option control plus enter, right? Here it is written run the current line or selection by using control plus enter. So this way you can execute. If you don't want this console, like there are multiple options if you execute and you just want to clean this, so you can use this option to clear this console. There is again a shortcuts control plus L. You can use this, or you can just click on this option. So in scripting, if we discuss, it allows in R base, it allows for execution through this plain text file. But there are no advanced code editing features. But here in R Studio, you'll get this powerful script editor like we execute here, with this all options like highlighting, completion, error checking is also there. Okay, so this is definitely enhance your code experience. Now console. So in R base, there is a console only where you interact with the software. But in R Studio, this console is nothing but your IDE, which provide more cohesive environment. So here, if you want a direct interaction with your terminal system's terminal, so you can work from R Studio with your terminals. Even you can check or run some background jobs if you are working with multiple projects. So at background you can execute any jobs and then work here for current work. This is all about console. Then let's move toward this. You can minimize or maximize according to your requirement. Like if you're working with some graphical representation, then if you need this fourth region, that is plot, so no need of this, so you can minimize this. When you don't want this area, you can minimize this like this, or you can customize like by scroll up and down. Then let's see this pane: environment, history, connections, and tutorial. Okay, so environment is basically, um, here we can check whatever objects are generated. If like I show this X, so whatever objects or variables, list, anything, data frame, whatever object is generated in this area, it is as a summary, it will store in this environment. Okay, so in current, our environment history will be there. Even this history, whatever the previous command we execute, we can check this. Then there is a connection. So connection allow for a different database or data sources we can import. And tutorial is for checking or for learning purpose. You can use this tab to know more about this R Studio. Then here, uh, first thing, if we want to set working directory, you have to use this command set working directory. In bracket, in quote, you have to write path, whatever the path. So if you want to check path or where exactly you are working right now, you can use this command get working directory, then execute. So this is get working directory and this is a current path, like this. So if you want to set any different path, then you can use this set working directory and get to know about the path. If I am not getting what exactly this code for, I want to write somewhere, so you can use comment. Like here, for comment you can use this hash to know current working directory or folder it is. If you want to write a multiple lines code, then there's an option like triple quotes, but it is not frequently used. You have to use this hash. Hash is just for our understanding, not for execution. Now,
Quick question: where is the path written after executing get wd command? Where is the path shown?
this path is basically like, okay, let's see. Suppose this is session. From session you can set your current working directory path, means whatever script we are executing, that script we can save in that our own directory. Like suppose I want to set a directory for your class, so I can set this working directory from this option, session set working directory. Sorry, my screen is visible.
No, it just stopped being visible.
Okay, just let me check, something happened.
Yeah, I was asking because I executed the command, and but I
Yes.
don't see the path anywhere.
Okay,
So I
okay,
don't
let
know.
me that one I am telling. Just wait, I think I close this somehow. So just go to that session, set working directory, and you can select any directory which you want to set as your current working directory.
Ma'am, your screen is not visible.
Yes, I'm working on that. Just wait.
Okay.
Okay, so I think it's visible now. So here you just go to session, then set working directory, then choose directory, and from here you can select the directory, current working directory which you want to work with, anything. Okay, as you see, suppose I select this CAD4, I'll select this as my current working directory. So this is your path. When I use set working directory, I can use here. Get working directory is to know current working path. If I execute this command, I'll get this path which I'm currently working. But if I want to set my directory, so I have to write by command line. Okay, if this is for command line, and if you want to use graphical user interface, then you can just click and select your path by this method. But if you want to write a proper path, so like this you have to write your path. Like you want to set this path, or let me create a directory, suppose like this. So similarly you can set path and then select that directory. Okay, set your directory. In this directory again you can create a day wise folder, day 1, day 2, day 3 if you want. Otherwise this is okay. So get working directory is to know current working directory, and set is to set current working directory. These two options are there. Okay. Now this also, if you don't want previously generated all objects, so you can just clear these all objects, clear objects from workspace. So include hidden objects. Hidden means like whichever generated by using dot or what. So that all objects, if you want to delete, you can just remove this history. Here you can check multiple packages, like as I told, for graphical view there is a package ggplot. So if I'll write gg, you'll get the all options available with gg. So this one is the package, we can work with that package. Okay, so this is the main benefit. No need to write everything through command line. You can use this IDE, and then we can just click and check. So this is all about script, console, workspace. Now in file, these are all files from current working directory. So you can select all, check all the files from current work selected for folder. Then plot, when we generate plot, you can visualize the plot in this area. You can also export your plot as a image with extension, then PDF file, or you can also copy to paste your current working document. Then help is to understand, suppose any function or any concept you are not aware about, any particular term, keywords, so you can take help of this tab. Documentation you'll get. Clear?
Ma'am, can you show me once again in the third pane, ma'am, from where you have cleared all the objects? Um,
Where? Third?
yes, ma'am.
This one. Here, this, everywhere there is a broom like image,
Yeah,
na,
yeah.
that is
Yeah.
for cleaning.
Okay.
When you generate plots also, na, you'll get this image. If you don't want to save this, so you can clear this from this current working pane. Now cursor is blinking here, suppose. So you can use shortcut control 1 and 2 option. Control 1 is for your script area, control 2 when switch to, means when typing, if you want to switch script area to console, console to script area, then you can use this option control plus 1 and control plus 2 simultaneously. Okay, so let's check, revise this like so you'll get a clear idea and you'll be comfortable with this environment. So we can write code here itself. So this is the first, that is script editor. Basically this is primary workplace. So here you can write, you can edit whatever your R code. So as I told, there are some features like syntax highlighting. Highlighting means whenever you write keyword, whenever you write variable, any comment, so it gives you a proper color notation. Like here you can see this object is generated, this value with the blue, object is with black color, then this is a path with green. Then whenever you'll generate some different data structures, for each structure there is a separate notation. Okay, so these all things available in this IDE R Studio. Code completion is there, error checking, even you'll get these line numbers. You can see here line numbers are there. Sometimes there is a code folding. When you learn this control structure and all, I'll tell you what exactly code folding and all these things, like collapsing sections are there. Okay, how to find and replace these all things are there. So this is all about script editor. Where console, basically it is for command execution, means interchangeably you can use this. Then here, there is a terminal, you can interact with your system terminal, right? Then background jobs is there. So what exactly the purpose of console? This is an interactive interface, so you can directly enter and execute R commands. That is the main thing. Then here in environment, basically environment is to check what all current history, current objects are generated in this script, in this current working area. So this environment is a tabular view. If you check here, as we have not created any object, let me create first. Okay, so here this is a table like structure which contains different objects, list of objects whatever we have created, along with name and the value. Fine. History to show previously executed command. Then there is a connection for connection database and data source. We can also import database, like here it is signed. We can save this, clear this, and then files to display the file directory or whatever the current navigation of your file management. Plots is for graphical visualization, and packages for installation management, installation loading, removal, everything we can do through this package tab. Help again is for documentation, and presentation or viewer for creating web based content, but this is not mostly used. Okay, so this is all about R base and R studio. Clear? So what exactly pros and cons for this R base and R studio? What do you think by observing this R base and R studio? Which one is easy for you? Just explore more so you'll get a clear idea. Okay, so R base is basically very lightweight version and fast. No need to use all these options. In one pane only we can work everything. But cause means like fault is what? There is a limited features, and uh, it is not beginners friendly because we have to write everything by ourselves. There is no code correction and these all options. Okay, but in R studio this is very user friendly. If you can see, just by clicking you can interact with R studio. There are many features for plotting, debugging. There are many options are available. Even built-in support is there. Clear? So but R studio, to execute R studio we need RBase. Okay, that's why I asked to install RBase first and then R studio. So in backend, ours today is executing for this execution interaction. Mm, I hope it's clear. Is there any query regarding?
No, thanks. Everything is clear.
Yeah. So from tomorrow onwards, we'll work like how to create object, how to create data type, data structure, and so on, which is basic syntax required to execute our program. Fine. So I'll stop here for today. Please mark your attendance.
Yes. Thanks for the session.
Yeah, welcome.
Thank you,
Okay.
ma'am.
Bye bye. Practice this. Be familiar with this environment so it is easy to understand.